Is there an apple tree in your backyard and you're wondering, what variety is this? Well, I have some good news for you. We're going to be hearing from Dr. Cameron Peace of the Washington State University on reconstructing the apple family tree and some cider genetics, which means basically they're figuring out how to identify apples via genetics. And my name is Rhea Wincaller. I'm both the producer and host of a weekly podcast called Cider Chat. Cider Chat is now in its 6th season with over 250 episodes, all archived and posted on this here YouTube channel. So what are you waiting for? Subscribe today. Just go ahead. Hit that button already. It's right there waiting for you. Now let's head to Dr. Cameron Peace's presentation that we have synced with his PowerPoint presentation, as he makes apple genetics easy for us to understand.
So I'm an associate professor at Washington State University. So I'll be talking about reconstructing the apple family tree, and I'll also throw in some cider genetic stuff into there as well, specifically on that. So some of the questions I'm going to be answering, hopefully, are these, at least in your own minds. Can DNA profiling technology be used to figure out the identity of any apple tree, or how any apple tree or variety is related to others? And how related are apple varieties, modern ones, and also when we add on heritage, how much more diversity do we get with heirloom varieties? And where did Honeycrisp get its ultra crispness from? It's really amazing texture. And what makes Honeycrisp so unusual? And is there any value in using crab apples in breeding? And is anyone bothering to do this? And when can we do 23andMe for apples? Right? And especially for your special tree that you've got, that you're really trying to find out what it is or what its origins are. Okay, so the 5 parts I'll be talking about are family trees and shared DNA segments. This will take up probably about at least half of what I'm going to be talking about today. And then I'll talk about the wild cider breeding program that we have at Washington State University. Then bring some of this stuff together about specifically on reconstructing the apple family tree, and then my apple, and people that are involved that helped really to put a lot of this stuff together behind the scenes, some postdocs at the front there, so Sten van der Zander and Nick Howard, and then some researchers from Europe, which I'll bring up later on as well, Eric and Chalarik, and also Gail Volk at the USDA in Fort Collins, who works with the USDA collection in Geneva a lot. Also the funding, so Washington State University and the Specialty Crop Research Initiative from USDA that funded a big project that provided a lot of the tools I'll be talking about here, Rosebreed, and also thanks to my own ancestors. These are just some of my recent ones, going back to my great-grandparents, and I weave some of them into the story too. So, okay, so first about family trees and shared DNA segments. So here's my paternal line, my father and my father's father and father's 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 father, etc. So at least up to Alfred Peace, who came to Tasmania in 1884, went specifically to Tasmania in Australia there, and his son, Lionel Peace, or Leo Peace, he was actually an apple grower, which I only found out a couple of years ago. My family decided to keep this to themselves, despite all the research and everything I was getting into, and not knowing why I fell into what I did. No one else is otherwise working on apples or anything like that. And so yeah, at Cunha, which is where he grew apples, and then his son was born, and then his son there was also born, which is my dad. Worked on stuff. So I mean, I'm interested to know what he grew and how he was involved in the industry there. So up my mother's line, up the maternal line, the one at the top there, Martha Watson, she came to Tasmania in 1885, and then her granddaughter, Violet Wiley, she was a teacher. She was one of the rare great-grandparents that I met. Here's me as a 2-year-old or so, behind her, walking around behind her, and I was carrying something, and then some folks took it off me, and it's from some stills I took from a little video that again, my family kept from me, and I finally came across this a couple of years ago, and it was an apple that I was carrying there. So the only video I have is a kid, and there I am with an apple. Building out to my bigger family tree, all various folks going up to, I think, my 4 times great-grandparents. I've got it over in the back there, and all various photos of all of those. I really want to know what I inherited from these folks and learn as much as I can about them. So we're now in the age of DNA-informed ancestry, right? So all these various companies that have these great products out there. Every one of these, I've got my DNA done with and compared and contrasted and done all sorts of fun stuff, as well as about 20 of my family members, most of which I've paid for. So there's lots of great tools that are out there for family tree verification and reconstruction. There's pie charts that you can do of regional ancestry. I don't think that's so useful, actually. It's a little bit interesting, especially if you've got some mixed ancestry, but otherwise it's not particularly useful. These other parts are pretty good, though. There's DNA matches with other people, and matches that then share with your own family tree. So cross-referencing those, and then chromosome browsers, which show where the DNA matches are that you have on your genome. So those, for family tree verification and reconstruction, are really powerful tools that these companies have. So yeah, quite useful and very useful for that last one, the chromosome browser. And that sort of stuff is what I'll be showing, is looking at chromosomes and where matches are. So DNA, of course, is a blueprint of life and coded instructions for all the great stuff that goes on in our bodies and any organism. Now these are arranged on chromosomes, just to remind you, and we inherit these from our parents. We inherit our DNA from our parents and get a chromosome each. So it's the same, absolutely the same with apples. So chromosome 1, for example, just to show here, so our chromosomes are actually paired, right? So as I was saying, one from each parent. Now we don't actually need to show it as X's. You always see it as X's, but it's just double the same information. It doesn't really mean much. So it's much easier to consider these just as linear things. And so humans have, of course, 23 pairs of chromosomes. Again, one from each of your parents. Then you can have, well, obviously there are genes all on the chromosomes. And so, for example, about 2/3 of the way down chromosome 1, there's a gene that influences extroversion versus introversion in humans. A typical view of chromosomes then will be this stacked thing. So like in 23andMe or something like that. Yeah, Ancestry is another popular one, but they don't have a chromosome browser, so they suck. They've got some other good tools. But they often display them like this. So you've got the 23 chromosomes, right? So 1 down to 22, and then this is a female here, one of my relatives. So there's two X's there. And in each case, the topmost one here is the upper bar there is what someone has inherited from their mother, and the bottom one from the father. I don't really like this stacked one. It's useful, but I've come up with a better method, a better way of visualizing, which is circular. Again, we've got the ring that you get from your mother, the stuff you get from your mother, and then what you get from your father as well. The inner ring is what you get from your mother. Outer ring is what would be inherited from the father. For example, just the outer ring is what I inherited from my father, and then colours match up with the various ancestors going back to even a piece that I know I inherited from a 5 times great-grandparent there. Obviously using some of those tools that I mentioned earlier that they have, and then some of my own genetic skills to get in and play with this stuff. So humans have 23 chromosomes, apples have 17. And we can look at these things in exactly the same way. So the reason why we can look at this and get this sort of information is because we now have these things called, there's this type of genetic assay that we can get and look at things called SNPs. It's been the key technological advance that allows this to happen. And so it allows us to look at information from thousands of genetic points all along each chromosome. And we can do this, and it's finally tipped us over the edge of being able to do the cool stuff we can. It's because of the really high density that we have. So we can genetically characterize any person or any apple tree, any apple variety. And the really useful thing is that it allows us to easily detect where one ancestor's DNA switches to another. So within the chromosomes, you've got the black bars, little black lines here representing the split between a chromosome. So that would be a chromosome. We probably are up to about chromosome 12 or something at that point. Let's go to chromosome 11 or so. So this switching between the two colours here is where it'll be some ancestor has, one ancestor has switched over to another ancestor. So we use SNP arrays, or some people call them SNP chips, and we have 3 available for Apple. We have an 8,000 one, so we call it the 8K array, and there's a 20K one, and then a huge one that's the same resolution effectively as for humans. I've got 20K one in green there because that's the one we typically use these days. It's the most useful and effective. The other one on the right-hand side's a bit of overkill. And so we can come along to plant tissue, and typically apple leaves is where we like to get it out of. Go there and we can check out the DNA and get these DNA profiles from the material that we run there. We've got some large data sets that have been developed in various projects in the US, the Rosebreed project that I was heavily involved in, a big project over in Europe, collected a whole bunch of material and lots of cultivars, right? Lots of cultivated varieties. Very recently, we just got in some data from about 300 Tasmanian, some other miscellaneous material as well. And then all this stuff, one of the guys I showed earlier, Nick Howard, he's been collecting all of this and putting it together. He's been adding a whole bunch of additional ones on as well. So he's really got the big master database there. So more than 2,600 unique cultivars, let alone many, many hundreds and thousands of seedlings, but actual cultivars from 24 collections around the world we've put together. And we've got some more stuff coming soon. It says 250 more US heritage cultivars, 600 more US heritage cultivars as well from various other projects that a bunch of us are involved in. So we can use this data, this DNA profile stuff to, especially for identity and relatedness. So we can easily use this, first of all, to detect redundancies in collections. And this comes up very often, that something in a collection has got it's got a different name as something else, and it came from another location, another origin, but it actually ends up being the same individual. Now, this comes up a fair bit. And I'll give some examples. Another major thing is parentage, and it pretty much across the board, whether it's varieties that are out there or seedlings or selections in breeding programs, about 15% of those records are wrong. Often there's no records and they're not wrong, we just have to figure out what they are. But where it has been recorded, quite often those things are wrong. Some new material that came in that was sent by John Bunker and friends there, and they sent me several individuals, some leaf tissue that I ran, and his book has just come out too, The Apples and the Art of Detection, which is a really great book. And in there, for example, is a couple of individuals. New Brunswicker is mentioned, and Nutting Bumpus. And you probably can't read the writing, but saying, hey, when I hear about these things, I assume they're probably correct as long as the apples have that Duchess, New Brunswicker, nutting look. He also says, where are the DNA testers when we need them? Wouldn't this be a great use of your hard-earned tax dollars? And if you look at those, they do look a lot like Duchess of Oldenburg. If you're familiar with the look of that, it looks like those. And sure enough, it came up that that's exactly what they are. They suspected maybe it's a seedling of or something, but no, they're actually just renamed Duchess, which has been around a long time. Another example was Cherryfield, which they realized also is the same as Collins. And then another one was sent in Benton Red, and it turns out that that's actually Salome. So not sure which came first in all that, who named which first. So some interesting history to look into that and try and figure that out. We've figured out that onto parentage, there's a whole lot of things that we've figured out. Since DNA profiling technology has come along, we've made lots of discoveries. So first of all, on the left-hand side of the column here, in green are the ones that have turned out to be correct. So Gala, for example, is Kids Orange Red, Cross Red Delicious, etc. Some of these other ones, Honeycrisp and Wealthy, incorrect. Granny Smith, also incorrect what that's been recorded as. So Wealthy, for example, is, if you look up the stuff online, you'll see, it's a Siberian crab descendant, and that's why it was cold hardy. Turns out not true. It gets it from Duchess of Oldenburg, is where it's just Jonathan cross Duchess of Oldenburg, and the cold hardiness comes from Duchess. For Granny Smith, it says to be revealed later this talk. Okay, so Braeburn, we now know, a chance seedling came up in New Zealand. It's actually Red Delicious cross Sturma Pippen. Golden Delicious, we know it's Grimes Golden. The other parent, we don't know exactly what that is, but we know Yellow Bellflower is one of its parents, so it's one of the grandparents, therefore, of Golden Delicious. We now know the parents of Red Delicious. It's Wine Sap and White Winter Pearmain. So all these amazing discoveries that we're coming up with here. And these are very recent discoveries, most of these things here, so we can now start to connect those up with the histories of all of those cultivars. Another example at the main heritage orchard there was Cranberry Island. This one was sent in, and so in the 2017 Common Grand County fair there was a bunch of things. Do you know these apples? Some people had provided some of these things. The Cranberry Island was one of these. So the samples were sent in to me, and so it ends up being Duchess of Oldenburg again, crossed with Zushoff, another old cultivar. And you can see it looks quite like that. In hindsight, all these things make perfect sense. But there's so many apple varieties out there that things tend to look the same after a while. So we can look at the relatedness among common cultivars. Let's have a look at that now. So let's consider Fuji, Red Delicious, and Gala. These are the top 3 produced cultivars in the US, but also in the world. They make up, at least 75% of everything that's produced. Might even be 90%, I don't know. Massive amounts. So are these related to each other? How are they related to each other? Well, here's how it is. Red Delicious is a parent of Fuji, and it's a grandparent of Gala. So then that makes Fuji half aunt of Gala. So there's a bunch of relatedness there. Let's add the other 5 top cultivars, at least in Washington State. Here they all are: Granny Smith, Golden Delicious, Crisp Pink, or Pink Lady, Honey Crisp, and Braeburn. So how are they related? Well, they are. I've basically put them in their spots where they all connect up with each other as well. So yeah, everything's related. Not everything's directly related to each other, but they all are part of a pedigree that we can reconstruct here. So Braeburn and Fuji are both kids of Red Delicious. That makes them half siblings because they share that one parent, etc. Honeycrisp has Golden Delicious as a grandparent, so that connects it in with lots and lots of things, etc. So yeah, we can reconstruct these pedigrees of cultivars. So there's a fair bit of relatedness here among common cultivars. So the way I see it is the stuff you get in the supermarket, those major cultivars there, it's like they're all in one street of a medieval village, right? So everyone's a close relation. What about heirloom cultivars when we incorporate all of those, right? That surely should add a whole bunch more diversity on. Well, a bit, not too much more. It's really just like we're still in the same village, we've just gone out to the rest of the village. And so you have some weird things. Over the creek there, you got old Duchess of Oldenburg, sort of who knows where she came from originally, but still she's got some grandkids and great-grandkids that are in the village there, etc. Others get pushed off to the side, but still everyone knows how they're related to everyone else here. So that's how it is. So heirloom cultivars still have a whole lot in common, and I'll show you some increasingly more and more connections through the various cultivars that we all know about. So we can look at this via shared DNA segments. So when we have bits of DNA that are in common, which I'll be showing with these green patches here, that means there are shared ancestors between individuals. And these are large chunks that we're looking at. So even the thinnest slivers that you see here of green, these are too large to be from 100 generations ago. This is 10, 5, 3, 2 generations away. So we can use the SNP arrays to do this, to find these shared segments, and the amount and patterns that we see really reveal what the actual relationships should be. So it's really great for pedigree reconstruction. So the picture on this side will look very much similar to this. So if myself compared to my mum, I've inherited half from my mum, and so it's the inner circle will be all lit up in green. It's just like Honeycrisp from its mother, Keepsake. When we compare Honeycrisp with Keepsake, then the entire inner circle there lights up in green. That's what it looks like when you compare a child to one of the parents. Up at chromosome 1, as I was showing before, about 2/3 of the way down is the extroversion stuff. It turns out I'm doubled up. I'm homozygous for extroversion, so I must have got one from dad and one from mum. But anyway, one from mum was up in there. If we take this up one more generation, up to the mothers of these mothers, we start to see these break up, right? So we expect what the original green ring there was, that should now be halving. So half of each of those chromosomes should be coming through from then on. So, the grandkid will inherit a quarter from the grandparent there, but it'll be half of that inner ring. As I was saying, so shared DNA comes from shared recent ancestors. So, for example, if we go back to two of my 5 times great-grandparents there, couple, Edward Gard and Hannah Green. It's expected that from any of our 5 times great-grandparents, any apple as well, that it's inherited 128th or 0.8% from each of those ancestors. Descendants then are inheriting at every generation a random half is what we get. Eventually it doesn't halve, so those little slivers that you see either are there or they're not there in a generation. So they don't just get smaller and smaller and smaller and slice in half. Once they get to about a quarter of a chromosome or so, actually they're either there or they're not there. So that can lead to some weird outcomes in terms of how you're related to someone. So here's someone that's also descended. Just a couple of months ago, I connected up with this person, Christine W. here, who's also she's a sixth cousin of mine because she's descended from the same 5 times great-grandparents. So it's expected that we would share from the half down there and the half that she inherited, that would only have 1 in 8,000 of our DNA, or 0.01% that we would share. Well, we share a lot more than that. We share 2 segments, which adds up to 0.24%, which is about 20 times what we'd expect. But that's not surprising, because otherwise, if we shared a tiny amount, even smaller than that, then I wouldn't have even detected her in the first place. We wouldn't have made the connection. So it's an example of some extreme that you can get, extreme variation from just whole segments being inherited or not. So if we look at these large shared DNA segments that we have among cultivars, let's look at Honeycrisp versus its half aunts, right? Crips Pink and Gala, through that grandparent of grandparent of Honeycrisp, Golden Delicious. So we'd expect that this half aunt, half niece situation, there'd be 12.5% or 1/8 of the DNA that's shared. So this is what that looks like. So the outer ring there is what Honeycrisp has inherited through that parent, MN1627, which then it shares with each of these cultivars here, Gala and Crips Pink. But you can also see there's a little bit shared on the inside too. So this is like, oh, hang on, that's through Honeycrisp's other parent. But still, it's about this. This 12.5% is about what you get here. And this extra part on the inside that Honeycrisp shares adds a bit more on as well. Takes it up to maybe 16-17% is what it's sharing. So if we look at Honeycrisp versus some other cousins that it's got, some half cousins, Harrelson and Fireside through their shared grandparent, Duchess of Oldenburg. So it's half cousin, it's also cousins twice removed as well. So we take it up through this other lineage as well. So through Honeycrisp's other parent, Duchess of Oldenburg also turns up as an ancestor. So in this case, we'd expect that Honeycrisp would share 1/16 plus 1/64, so about 8% with both Harrelson and Fireside. And here's how it looks. That's what those sorts of proportions and the patterns look like for these sorts of relationships that we have here. Then we can take a couple of really famous cultivars, which I'm about to show you in a minute, cultivar A and B for now, compared to Granny Smith. This is how much they share, but there's no known pedigree connections between them. And yet it's the same sort of patterns as what you were just seeing before, where we did have those known connections. So they share 15 to 17% of their genome in these patterns with Granny Smith. So that's equal to at least first cousins, and yet we don't know the actual relationship that's doing that. And that's Red Delicious and Golden Delicious. So there's a lot of these extra connections that there are. Another one is Honeycrisp. If we look at Honeycrisp and compare that with some crab apples, it turned out that there's a couple of segments in the Honeycrisp genome, total of 1.3% of its genome, adding up in these 2 segments, that match with nothing else except crabapple. So it turns out that Honeycrisp actually has crabapple ancestor. So it got it through frostbite. If we look at frostbite, has 4 times as much effectively than what Honeycrisp has. It's just halved and halved as it came down. And so we can look at the segments. There's a whole bunch more. There's 7 segments there in frostbite. And so we can deduce that there's another 3 or 4 generations, 2 or 3 generations intervening between frostbite and some other individual up there, which is probably a Malus robusta, similar to Dolgo, the pollinizer. And so what this means is Honeycrisp has a wild 3 to 4 times great-grandparent. Frostbite's really unusual in its characteristics. So the University of Minnesota that released this cultivar says biting into this apple is like biting into tropical sugarcane. It's one of the most distinctive and unusual apples we've ever tried. It has aromatic, crisp, crystalline flesh. Right, so tropical sugarcane is not a typical thing typical flavour described for apples, but you see it more and more descriptions. I've seen it actually in various other crab apples as well. And I think the actual original species, prunifolia, I keep coming across that one. Keepsake is hard and crisp with an unusual flavour that's somewhat like sugarcane, they said. So there we go. So you can see Keepsake has inherited some of those attributes, those very unusual attributes that Frostbite has. And then Honeycrisp, well, Honeycrisp doesn't have that sugarcaney flavour particularly, so it seems like it's lost that. But of course, it's got this ultra crispness, so explosively crisp, as the University of Minnesota says, and this makes Honeycrisp so unusual. A crisp, juicy texture that exists in no other commercially available apple variety, except for now kids of that, which are being released. So it's the most important breeding parent in the US for the last 20, 30 years. And the rest of the world's catching up now with this, taking on this attribute of this ultra crispness, which attracts a large premium at retail. And so basically we've figured out where that came from. It didn't come from just normal old things that ancestors, that bits and pieces have come together. It really looks like it's because of this serendipitous wild species background. So here are the two segments. We think both of those are required, that within there somewhere are the genetic factors 1 and 2 for providing that ultra crispness. We think both are required. But some other things have come along as well, some good and some not so useful. We've got high acidity, low tannins, low browning, bitter pit susceptibility have come along with the one that's up on chromosome 16. And then the piece that's at the end of chromosome 6 there's a good thing that's come along with that one, 5 light tolerance. Looks like it's also hitched a ride, so that's a bonus. So let's look at another case of wild species coming into apple. So 80 years ago, scab resistance breeding was done, purposely using the tiny fruited Malus floribunda crabapple at the PRI breeding program. And here's an example of one of the cultivars that's come out, Enterprise. There's been a lot of other scab-resistant cultivars came out of this program, many of which you'll know of. And they all therefore have that scab resistance is coming from this wild ancestor. So it took 6 generations to get to something that they could release eventually here in the terms of Enterprise. It's a similar number of generations to get to the various other cultivars that there are as well. Okay, so it starts up with floribunda, and then we're expecting that wild DNA to be halving as we come down through each generation. Right, so well, we can use the same tools as before to look at these. So ultimately we expect 3.3% is what we'd get in Enterprise, is what we'd expect it to come down to. So we can visualize it. Floribunda itself is completely green if we use the color green for this species. It's 100% itself. It's kid that was crossed with Rome Beauty, that descended from a cross with Rome Beauty, therefore will be green. Entirely there will be 50%, and it comes on through, and this is what it looks like as things come through the generations as we track this floribunda DNA all the way down. I'm just showing here, this last one here is just one of the parents. The other one, COP7, is at the same level. I'm just not showing that one. But then Enterprise ends up having 4.4%. So it got one of the pieces of floribunda from this parent, and then, which is over here, which is actually where the scab resistance is embedded inside there, and then the other one's on the outer ring there, the other 4 segments there, that's what it's got from its other parent, COP7. So what about if we combine these wild species? Put this floribunda from something like Enterprise has got these pieces, and Honeycrisp has got these, and they're in the standard species, right, Malus domestica in that background. You know, what if we combine those together? So that's being done, and that's what Cosmic Crisp is, or WA38, as we fondly know it, at Washington State University. So this is commercially released recently, and it'll be available in the supermarkets apparently this month, all across the US. If we look then at the genome of Cosmic Crisp, it has inherited both of those segments of Robusta that I got from Honeycrisp on that outer ring. And that's why, well, one of the reasons why we think that both pieces are needed to make ultra crispness. And then it got 3 of the segments of Floribunda from Enterprise. It turns out that it didn't get the scab resistance though. So this little sliver up here is in almost the right spot, but there's actual Enterprise has a little bit more green. So the actual gene for scab resistance is just into the grey. And so just by chance, it split right there, recombined right there in the ovule that became WA38 and lost, well, didn't lose, but did not inherit the scab resistance. So this is the sort of thing that modern breeders are doing, right? Is incorporating all this wild stuff and various ancestry coming into things. So it's not all this inbreeding and everything that everyone typically thinks it is. There is a lot of diversity incorporated into modern cultivars. If we take this even further and look at all the ancestry of going back through the pedigree of WA38, then we can colour it all up, and that's that majorly coloured diagram that you see before. So all the various ancestors through Honeycrisp, so WA38's father Honeycrisp on the outer ring, and then all the various ones on the inside there as well. And the yellow here, Golden Delicious is in the ancestry, the pedigree of both sides of WA38, both through its mother and its father. And we can put a whole bunch of genetic factors on as well, because we know a whole bunch of genes that are controlling various traits. So there's the stuff I was talking about before about the ultra crispness and fire blight, resistance and high acidity, and then didn't get the VS scab resistance, so that's in red, like green for good and red for bad, etc. And then more things we can put on. So this is from the extra dark skin that WA38 has. It's because of the 2 copies of the blush genetic factor there. So it's highly blush, just like Enterprise is, but also like Jonathan. There's not too many cultivars that have that. And therefore we can track the origins of any of these. So we can look at any of these and go, oh, where did it get its favourable allele for firmness from? It's got it from Duchess of Oldenburg, that particular one. And then it's got its other one from Red Delicious. And then we can track that back, right, behind Red Delicious back to whether it's the White Winter Pearmain or Winesap. We can look at the diversity of any cultivar, so we can look at what new alleles, what new genetic factors Cosmic Crisp brings to commercial production by comparing it with the top 8 Washington-grown cultivars here. So here's WA38 compared to Red Delicious, shares 20% of its genome. Let's go through each of these here, shares all these large amounts with all these other cultivars out there. Then we can do a subtractive analysis and say, well, what does it have that it doesn't share with any of those? And this is what it is. And it could be some of those Floribunda segments. So 6% of what it has is new, and that brings, injection of new diversity. It helps resist stresses, that sort of thing, in production. So that's one way of looking at a new way that we can look at diversity now. Okay, on to the second part then. The WSU's wild cider breeding program that we have. So just a bunch of pictures just to quickly introduce you to this. So we've got this breeding program going on for experiential learning. Students run it, they pass it on to more students as students come and go. We started this quite a few years ago. This is planting that was actually done back in 2010 here. So I got this going as a hands-on opportunity for students, but also mouth-on, most important. So here there are a bunch of my current students out in the orchard about a month ago. A key ingredient that we have in this breeding program is the massive diversity that we have. So we have on hand more than 20 of the Malus species, and that's why we've got the word wild in the title of the breeding program, which brings all sorts of genetic factors in and mixes all sorts of things up. So it's a load of fun. The first class in 2014 decided that they wanted to breed for cider. So here's all the spitballing on the page, on the whiteboard there, and then they unanimously agreed, yeah, let's do hard cider. So excellent, everyone got behind that. They came up with all the things to breed for then. So they create and advance new germplasm each year, move it all forward. So lots of grads and undergrads, interns, volunteers have been involved in this. Here's folks crossing through the years. So every spring we do a whole bunch of crosses, make several thousand seedlings or seeds, and then grow those up and eventually plant out maybe 1,500 or so seedlings in the greenhouse. Although we end up throwing out a whole bunch too, so we might germinate the seeds, and then on this picture up here is just throwing out. It's all 1,000 or 2,000 that we threw out for certain reasons, purposeful reasons. And they look after these. Lots of diversity we have. And then a whole bunch of those get thrown out. So here's Hannah here just throwing away stuff that we decided was not good. Again, proper reasons for why we're throwing this stuff out, but a lot of selection going on to whittle things down. We have two major locations where we have our material. One is out in one of the orchards near. They're both right next to the university, but one's out in the field, and then we're moving things to, especially all the seedlings now, growing them up in large pots. That's our new system to make things a whole lot easier. This is a photo I took last week of the material in the pots there. And trees, whether they're in the pots or out in the field, get evaluated all the time by the students to figure out the various tree attributes. There's certain things that we want them to look like. And then there's fruiting as well. So the fruit are also evaluated. Big Blush is, two of the photos here, is one of the seedlings that produced fruit after just 2 years after being planted. And it had 5 other ones from that very first lot that we had that also were fruiting really early, so that short juvenility, we're very excited about that. We've got a lot of material we use for red flesh, so we have that in there too. We're already on to producing the grandchildren of Big Blush as well, so that's how quickly we're getting through the generations, thanks to using wild species, as Malus sieversii is half of what Big Blush is. And of course we're making cider too, so this is from last week. A whole bunch of our selections. We have bitter bomb and bitter cream and bitter grenade and stuff like this is what we call these things, and making cider out of it. Pink Puma, another one there. So that's a whole lot of fun. So the target attributes that the students decided to go for, essential ones, they need to be high in tannins, so bitter but not being astringent, fire blight tolerant, at least tolerant, annual bearing, short juvenility at least by their 4th year, a certain size and certain juiciness, and be high yielding and mildew resistant. Whenever we see any seedling that has any mildew on it, which probably most of these do here that they're throwing away, it gets tossed. Throw away anything if it doesn't have all of these. If it ever shows that it doesn't have one of those things, we throw it. Then there's a bunch of attributes that are enhancing. So it gets extra attention if it has any of these things on the right-hand side. So it's even higher in tannins. Again, low astringency, but high bitterness. If it's fire blight resistant, excellent. If it's a red flesh, bonus. These are all bonuses, basically. Even shorter juvenility, etc. So that's the characteristics, the thresholds, etc. that the students go for. So if we bring it back to pedigrees again, then I was talking about Big Blush before. I said it's half sieversii, and it is. So we know its mother, and then we know where its mother's mother, and we also have a really good idea of the maternal grandfather as well, where that came from. So in Kazakhstan, and a particular population in Kazakhstan that was collected by USDA researchers. That's where that's come from. But the Big Blush is larger than its mother. Its mother's only maybe this big and green, yellowish green skin. Big Blush was clearly blushed and larger, which is why we call it big, even though it's only that big. It's like a small gala. But through genome scanning, we did the 20K SNP array on it, and we found out, Harrelson was the father. And we did have Harrelson growing nearby the mother, and 200 or 300 other cultivars, so we got lucky that we found what it was. It could have been one of the ones that we haven't DNA profiled yet. And then we know the parentage of Harrelson, etc. We know even back behind Jonathan, which is what the dots are there. And then, as I said, we've used this stuff to cross further. So, we've gone to the next generation, crossing Big Blush with another thing we call Red 2. We created Red 2. Red 2 was from Robert's Crab Cross, Novosibirski Sweet. We know some ancestry up behind those too. So we're already moving on to next generations. We're on to the grandkids, actually, as I was saying, of Big Blush now. So, reconstructing the apple family tree in general. Well, I want to acknowledge a few people that are involved again. So Eric van der Weg from Wageningen University in the Netherlands, Charles-Eric Jurel from INRA in France. Love doing this sort of stuff. As Eric said to me recently, this topic is my big love. He said, Nick Howard he's the one collating all this information. He's over in Germany right now, about to go work at Wageningen. But he was a postdoc from University of Minnesota and really got excited. He's the first author of the paper about the Honeycrisp pedigree that we reconstructed recently. And of course, I love all this stuff. So let me give you the pedigree of Granny Smith. So with a little story that goes along with it, right? So if you look it up on the internet, etc., then you find out it was discovered apparently in the 1860s in Eastwood in Sydney, which is in New South Wales. It came up as a chance seedling, right? So it was Granny Smith and did it. So in 1924, this is where the story goes back to the best account we have is from 1924, that the seedling had developed from the remains of some French crab apples grown in Tasmania. Well, first of all, French crab apples, it's not like it's a crab apple from France, it's French crab. The C should be a capital C. It's an actual cultivar that was really popular, and yeah, it was grown in Tasmania. It was a major one that was grown in Tasmania. So that indicates then, okay, French crab, which is meant to be have sylvestris in it, hasn't been quite confirmed yet, but could be this sylvestris, a European crab apple species, and then something. Then what's the father? So it would have been something else growing in Tasmania around the 1860s. So one of the common dessert apple cultivars there. Now, Granny Smith is particularly important because it turns out that it is, but the parent of Lady Williams, and then Lady Williams is the mother of Cripps Pink, or Pink Lady, so also very important, and Cripps Red Sundowner is another popular cultivar in Australia at least, and the other parent there is Golden Delicious. So Lady Williams' other parent is said to be Ropewood. That's what the records say. So it turns out that yes, Granny Smith is the parent of Lady Williams, and Lady Williams is the parent of Pink Lady and Cripps Red. So it turns out instead that the other parent of Lady Williams is a cultivar called Yates, which was brought into Tasmania quite a long time ago. And now we know the parents of Granny Smith. It ends up being Cleopatra, also known as Ortley, and Stone Pippen is another cultivar there. And I'll talk a little bit more about these ones. But when you look at them, and I have looked, and this is me holding Cleopatra and Ortley at the collection in Geneva. And it's like, why didn't people say this is the parent of Granny Smith? It's so obvious. And you bite it. And then when we found out very recently what the other parent was, and we look at what Stone Pippen looks like, it's like, oh my God, this is too obvious. So very recently, I've got a whole bunch of data on the pedigree of these Tasmanian cultivars, the down under apples, which is the basis of the Australian industry, and also a lot of New Zealand stuff came from here as well. So popular in the 1850s were these 7 cultivars here. The first 3 are the ones I'm really going to focus on because it ends up the other 4 don't matter. They haven't really come into anything here. So Stone Pippen, Scarlet Pearmain, Alexander. Okay, they're already growing in orchards in Tasmania, and Tasmania was the apple isle. That's where Australia's apple industry really went crazy and took off and supplied most of the apples for the country and for England, just like Washington State does for the US. By several decades later, some new cultivars had come in: Cleopatra, Sturma Pippen, Scarlet Nonpareil, and Adams Pearmain. So just the first 3 and the first 3 are what I'm going to focus on. Again, that the other ones don't seem to have come into anything. So if we take the first 3 that are at the top before, and then a whole bunch of cultivars that they have in the collection, this collection called Grove in Tasmania. Checked out, they're just names on a list, usually in alphabetical order, which drives me crazy because I know there's more structure to collections than just this alphabetical list of things. And so that's what we're doing, is really reconstructing the structure, the genetic structure of these things. So it turns out that Dunseedling, Nubina, Ranelagh, kids of 2 of those early cultivars, right? So Stone Pippen, Scarlet Pearmain. And these other ones also kids of 2 others, including Democrat, which is an important one that came out of Tasmania, ends up being that is Scarlet Pearmain and Alexander. These are all discoveries made in the last couple of weeks, by the way. I already told you about Cleopatra and Stone Pippen being the parents of Granny Smith. Then if we look at Cleopatra crossed with Alexander, it comes up with a cultivar called Prince Alfred, that another Tasmanian one. And Granny Smith and Democrat, Brown's Apple and Blanchard, a couple more cultivars. And then Red Delicious started to come in the early 1900s into Tasmania. And so, of course, more cultivars that were developed and named in Australia and Tasmania. You start to imagine that Red Delicious starts to come in as one of the parents. And sure enough, 2 of them there from Granny Smith with that. And then Democrat and Red Delicious came out with a cultivar Delicrat and Lagana. I thought that was a misspelling when I first saw it. And then we ran it, and it came up as the parents. They're the 2 parents, and then I went, very smart. They knew the parents. Well done. Very good. Maybe that particular orchard only had those 2 cultivars in there, so they could be pretty confident about it. Or maybe it just looks really clearly like it's a kid of those 2. Then Famoose, or Snow, or as they called it in Tasmania, Ladian Snow, came in, crossed with Scarlet Pearmain to give a cultivar called Crofton. Actually, it's a really important one. It ties in with my family, actually, this Crofton one. Then Sturma Pippen was another one that I had mentioned, came in, and so some more stuff came out of that. And Sturma Pippen crossed with Red Delicious is where Braeburn came from. So all this stuff all starts to get all connected up nicely here. So if we take Scarlet Pearmain, if I just go back, there was Scarlet Pearmain up in the top there. We can go back even further, because it turns out Scarlet Pearmain, we know its parent, and that's Scarlet Nonpareil, which is also a famous cultivar in Tasmania, not from Tasmania. But it turns out that Scarlet Nonpareil that's in Tasmania is actually Reynette de Holland. And then that one is a kid of French Reynette. We also know the other parent of it, but French Reynette, that's been a really, really important one, because it turns out that French Reynette pretty much is the grandmother of everything. That's the way we see it these days. So here's Charles-Éric Durel, talked in a conference, and this is me taking a photo of his slide in China last year, and then he said, yeah, you can use it. So, but it turns out, yeah, Reynette Franche, as he says, because he's French, but French Reynette comes up as being just so many things are descended or a grandkid from there. And I'll bring that one up again. So let's look at Jonathan, Golden Delicious, Cox's Orange Pippin. How are they related to each other? So if we look at their DNA and how they're shared, and then I've added a bunch of other cultivars on here are the patterns that they look like. Okay, they're large segments of DNA that they share. So when you see a pattern like this would indicate that Jonathan and Golden Delicious, this is 23% of their DNA, and there's the patterns. This looks like it's the equivalent of them sharing all 4 grandparents. And this pattern with Granny Smith, 3 grandparents, 2 grandparents is with Golden Delicious, Cox's Orange Pippin. It's what it looks like. Right? We don't actually necessarily know these connections, but we're getting some hints now. But this is what we can already start to imagine, figure out. Okay, even this 5% that Golden Delicious shares with these other ones on the right-hand side here indicates that, and in those patterns, that's 2 great-grandparents that they share. So we can take those inferences to come up with inferred ancestors. We can put on what we know, which are these solid lines, and even put on some of the cultivars that we know. And going back to French Reynette up there, so we do know that East Spitzenberg, for example, comes from there, and you can see a bunch of these things here. One of the grandparents I said of Golden Delicious before is Yellow Bellflower, and a grandparent of Yellow Bellflower is French Reynette, etc. And then the dotted lines are what would be required to make it so that these things become effectively true, or at least that level. So yeah, so we've got a bunch of inferred ancestors. So what we're hoping is that as we collect more and more tissue and people send stuff in, that we find these missing ancestors. In fact, in the Tasmanian collection, there was one individual came up that it's not in this pedigree, but it came up as one of the major ancestors, was an inferred thing, that a whole lot of things come off, and here it is, we found it. So we can place newly tested samples then. So let's say out at Steptoe Butte here with Dave Benscott, he's a tree that he's saying here's this old thing that's been out here for a long time. We could take the tissue from there, test it out and find out, for example, oh, it is that individual. Wow, it fits into the pedigree right there. That would be amazing. Very often we'll find that it's a kid of a couple of other things too, which also is helpful, but at least it lets people know what's going on. So that would be a lot of roadside trees Creekside trees will be that sort of thing. It'll be just a seedling of a couple of known things in the database. It's useful if it's a kid of something that is still inferred, because that gives us more genomic information about that to help reconstruct that missing individual even more. So I think that we can put everything into a single apple family tree. So my hypothesis is that all existing ancestors of all those ones on the outer ring that we don't know their parents of cultivated apple are related to each other within a handful of generations. And we have the opportunity now, with the tools that are available, to reconstruct this. I think we can put together the entire pedigree of cultivated apple, whether it's dessert apples or cider apples. They're all interconnected. So I think we can ultimately develop something like this. It's just a hypothetical, but one big happy family tree, right? This is really what it ends up looking like. Be more like that. So that's what I'm working on right now, is to redo all of this. So that leads me on to one of the initiatives, My Apple Tree, as we call it. So this is where you can add your apple to the apple family tree. So it could be any of your things that you have in your backyard or your grandfather's tree or whatever it is, and we can figure out where it is, where it fits in to the whole system here. As I was saying, I'm hopeful that we can find some of them that end up being ancestors. That's my interest in it, but otherwise giving answers to people about what they have. So the steps to do this, one is to get some leaf tissue and get it into a tube. I've got tubes here. Next is to mail it to us, to me, to my lab. $100 is what it costs at the moment. Wait a bunch of time, unfortunately. And then with your email address, you get your report emailed to you, is the plan. So this is actually available now, thanks to the technology that we developed in the Rosebreed project. What I'm hoping to do is make a cheaper, shorter, simpler alternative. That only costs like $10 and only takes, let's say, a month or whatever. This is what I'm trying to do now. So that would be simple things of check if one sample matches another sample. You send in 2 samples and we can just see, do they match, whatever they are. And then the second one would be, is something a kid of something else. If you want to know more, like how it all fits into the pedigree, then you'd have to go for the bigger one, the $100 one. So this sort of test then would be useful for, you got some weird orchard tree, that row, it's all meant to be the same thing, but there's one that stands out, what's going on, or several trees along the creek or the fence, etc. Or testing trueness to type before signs are distributed. There's a big issue with that, I know, in the cider industry. So that's what I want to get this available. So it's not yet available because it needs new research project funding for this dedicated effort. So if you're interested in getting your stuff done, then I'd say first of all get yourself tested on one of the companies, because it's so much easier to understand all the rest when you really feel it. I really got all these ideas from doing the human stuff. Get the tubes and instructions from me. I've brought both of those now. Otherwise, you can contact me. Collect your leaf sample, send it in, send your check, wait patiently, and then discover what you have. And that is the end of my talk.